Hey, welcome to day 3 of summer internship in phytomicrobiology. So you have seen rate of settling of various airborne bacteria in the last presentation. So we have used the same bacteria for practicing gram staining because the endophytic inoculation is happening parallelly. It consumes time for initial endophytes to come out. So we had a small theory session explaining how to do gram staining, and I would say the results are very impressive. The ultimate objective of any staining in microbiology is to have a isolated cells, single bacterial cells, or single ability to see single cells very clearly. Okay, I guess in this picture it is very much achieved. Okay, so we did a gram staining, and I will also say you can see an endospore inside this. Most uh, possibly this is bacillus. You can very clearly see an endospore inside this. Okay, why gram staining sometimes reveals endospore? It is possible. The crystal violet is actually staining. This is gram-positive bacteria. The peptidoglycan around the cell wall has been stained. When they are producing endospores, the endospores bulges. So the cell wall that is immediately after the endospore wall is actually very thin. They will have a thin peptidoglycan layer, usually results in less absorption of diet. So you will see in the bacillus, the center portion is oval and the polar regions are stained. So they are not actually designed to specifically stain endospores. They can sometimes indirectly visualize, endospores can be visualized using gram staining like this. So the what's happening here is endospores have a tough coat, keratin-like coat, and a dense peptidoglycan. The endospores may made up of the keratin-like protein and make them very highly resistant to chemicals. But during gram staining, the crystal violet do not penetrate the cell wall. So endospores are not stained, but the vegetative cell stained in purple, but the endospore remain unstained, will give you something like this. Yes, this is central spores, I would say. The spores are very clearly visible at the center, and the flanking regions, they create a halo effect. Okay, halo effect is very much seen here. I would say in this picture, the objective is met, of gram staining is clearly met, but still the contrast can be increased. Okay. And when you have such a picture, it is a very much indication that you may be working with the actinobacteria. See a filamentous structures that you can observe. Maybe this is an early stage of actinobacterial growth. Okay. The image clearly shows long thread-like structures with branching patterns. You see the hallmark of actinobacteria because they produce a submerged mycelia, and when they want to produce an aerial mycelia, they will decay itself. They will degrade its submerged mycelia, produce antibiotics, then start building aerial mycelia. So they will have a filamentous-like structure. That's why when these guys are discovered for first time, they are called as actinomycetes for a long time, until unless Carl's classification and many other people proved that their 16S RNA sequences closely resemble bacteria. So now they are, sorry, we know it's an prokaryotic organism. Okay. Um, it's a diagnostic cue also. Whenever you do a gram staining, when you see a filamentous bacteria like this, it's actually a diagnostic cue that it is an actinobacteria. Okay. So this is also a perfect picture of individual cells clearly dispersed, and also I can see endospores that have been easily located inside the cell. Okay, these are very well taken picture. Similar here, I guess the voltage of the lamp can be little more adjusted, and the resolution, I mean the contrast or the sharpness around the individual cell can be worked on. Okay. Definitely the contrast need to be worked on. They looks like a cockeye, but we need to work on the contrast. How to know or how to increase the contrast? This is definitely a cockeye, but still we can work on the contrast. I will tell you how to increase the contrast in few slides. This is a perfect picture, I would say. Perfect gram staining picture. Okay. You see, everything about this picture is okay, and it is also mostly like because the volume of gram positive rods here is actually much more thicker. So they are easily, excellent contrast is there. The gram-positive rods are vividly stained and showing strong purple coloration while the background remains clean and unstained. Okay, this is why it is having enhanced visibility. And see the crisp focus. The image is sharply focused at the high magnification, showing very clear cellular boundaries. Can you see that? And even internal granularity is, details. If you can capture more details, it is more elegant. Okay, you can even see the internal granularity, which is ideal for microscopy documentation. Okay, even staining, there is a uniform coloration across all the cells, indicating proper timing that is being followed during gram staining, and no over decolorization or understaining. It's perfect. All right. So the cells are neither too crowded nor too sparse, allowing each observation of the morphology and arrangement without overlapping artifacts. Maybe little overlapping here, but see in other areas, meaning the smear has been prepared very precisely. And the field is clean and minimal background noise. That's what makes this very visually appealing. And the rods appear distinct and separate, helpful for identifying the shape. And that's why we need to have individual cells clearly visible, because we need to identify the shape and size. All right. Even here, I could say an ideal example of rods in chains could be an actinobacteria in early stages. We have to wait and see. Okay, I think still contrast has to be increased. Definitely contrast need to be increased. Again, another cue that this could be an actinobacteria, because you are seeing a filament-like structures. Okay, this can also be an actinobacteria. Another important character of actinobacteria is when you try to sample the bacterial colony, it is not easy to sample. If you struggle, it's not like bacteria. E. coli, you will touch upon, if you touch the inoculation loop over the colony, it will just it will get stick down to the inoculation loop. But actinobacteria, you have to literally scrap it off from the agar, because they produce a submerged mycelia. It has to be scrapped off. Then that's how you can sample them. So when you have a difficulty while sampling a bacteria, have a suspicion that it could be an actinobacteria. When you observe a filamentous-like structure, it is definitely an actinobacteria. And this is non-sporing rods. Anyway, sporulation means you have to go for spore staining to confirm whether it's sporulative or non-sporulative. But I could say they are slender rods, no halo effect is seen here. I feel the contrast of the image of individual cells can be increased. How to do that? Contrast should be increased, but objective is met. And here also we have centralized spores. This picture is also ideal picture. Usually, see, all these pictures are taken from mobile phone, observed through the eyepiece. Okay, so usually the surrounding area is always blur. Okay, you have to trim the area out. You can see the circumference of the field view is always blur. Only the core region is sharp. Just trim it out and use that. I will show you few trims I have did. So see, here is another proof the mobile phone is used. See, we have this is seen through the mobile phone through the eyepiece. We have the black areas here. Better you trim it. Okay, trim this region and have only the core region. They have sub-terminal spores. Okay, they did not take the dye much, but few cells are stained better. Here also we can have the core region trimmed out. They have a sub-terminal spores. How am I saying it is a sub-terminal? You see the spore location of the spore in the individual cells, not like previous bacillus. It was little towards a polar region, right? So this is an ideal picture showing that it actually you have a contaminated culture. Even though you try to pick it from single colony, this is not a single colony. Okay. Seeing you have differentially dye-absorbed cells in this field of view, meaning the cell wall composition, even though you subjected every smear, it's the same smear, right? For same duration of time, there is a differential absorption of crystalloid. That's the first indication that something is wrong. And the second indication is, you see these two cells here, they don't have, they don't seems to have any endospore within them, but I can see spongily stained endospores lying around, meaning they definitely don't belongs to same species. How come an spore forming and non-spore forming vegetative cells can be there in the smear? Okay, so Objective is met, individual cells are very clearly captured, but the contrast should be increased. Here, this is not a contamination. Many microbiology students will presume that this might be a contamination, but it is not. You see, whichever cell that is been spongily stained or weakly stained is actually an endospore. I should call them as an exospores because they don't have a vegetative structure now. So these are exospores. The endospore forming vegetative cells are here. Whichever cell is been thickly stained as a purple, they are actually an endospore cells. And the vegetative structures that got disintegrated, leaving the endospore in the environment, now called as an exospore, are actually weakly stained. Okay. Contrast can be increased, the sharpness of the image can be increased. I think too much dye or the volt, or I think the diaphragm, we need to work on the diaphragm. Okay. If you have worked on diaphragm, you'll have much better picture. Okay. And contrast can be increased. So how the contrast should be increased, I will tell you now. Okay. You see, you know the various features of microscope. The option number 8. You know this is coarse fine adjustment, which can move the stage up and down, and this is a lateral movement of the slides. But you should know 3 adjustment. Okay. Here is also a stage below the stage, condensed stage that can be moved close to the slide or moved away from the slide. Okay. And we have a diaphragm to control the intensity of light here. We will also have one more knob on the handle, probably that side it is there. It is a voltage knob that can regulate the voltage. So use by, in my experience, when I keep this stage, diaphragm stage very closer to the stage, there could be more intensity of light coming in, but the contrast is much better. Now I can control the intensity of the light with the diaphragm adjustment. Then I will work on the voltage, and voila, you will have a very precise cells to be documented like this. Okay, so contrast can be increased. Very good. These are the clippings I have trimmed out. Okay, you don't need to keep the entire trimmed picture. Make sure all the dull cells or contrastless cells are removed. How to remove this? Just focus on the core region of the view. You will have cells with better sharpness. Okay, always at the core region. This is what I told. Using gram staining, how to find whether my colony is contaminated or not. You see here. These are actually non-spore forming. Sorry, these are actually spores, or a vegetative cell that is about to release the spores. Here we have a non-spore formers in the same. Okay. So this is not contamination. See, this is the vegetative cells. All white color circles are vegetative cells, and all orange color are actually exospores that spongily took the dye. So that's what happening in the lab. Very happy that the interns can pick this in even from the day one. We are inaugurating samples today. I will update you with the results tomorrow.